Hi semua. Now I will share some things about the regulation and expression of genes involved in the Lac Operon. And I will start with the concept of operon. What is an operon? It is a control mechanism for a cluster of bacterial genes in which the genes will share one promoter and will be expressed as one single unit of transcription. The bacteria can control their metabolisms, and this involves an operator, and this will be based on their environmental changes. This control mechanism is important for the bacteria because they are small organisms in which they need to conserve their energy, they need to conserve their resources. And so if there is no need for the bacteria to produce a type of protein, then they will no need for any transcription and translation. There are many types of operon actually for the bacteria, but one example that we have to know will be the Lac Operon. So what is the Lac Operon? This is the regulation for the expression of these three genes in the bacteria known as E. coli. The three genes that are involved will be the LacZ gene, the LacY gene, and the LacA gene. The three genes will code for three important enzymes that will be involved in the metabolism of lactose. And the three genes will be expressed together as one single transcription unit. And the enzymes will be synthesized only when lactose is present. But before we talk about how the enzymes can be produced when lactose is present, let's look at the components of the Lac Operon. Component number one will be the promoter. This is important as the binding site for the RNA polymerase. You should remember that RNA polymerase is the enzyme that is important for the process of transcription. Component number two will be the operator. This will be the binding site for a particular type of protein that we call as the repressor protein. And the operator can sometimes be regarded as the switch for the operon. Now, component number three will be the structural genes, in which this consists of three genes known as the LacZ gene, the LacY gene, and also the LacA gene. The LacZ gene codes for a particular type of enzyme known as the beta-galactosidase. This enzyme is important in catalyzing the hydrolysis of lactose, and lactose will be hydrolyzed to produce galactose and glucose. While LacY gene is the gene that codes for the enzyme permease. This enzyme catalyzes the transport of lactose into the bacterial cell. And the LacA gene codes for another enzyme known as the trans-acetylase. This enzyme catalyzes the detoxification of other molecules that entering the cell through the permease. So you can see the Lac Operon consists of the promoter, the operator, and the three structural genes over here, the LacZ gene, the LacY gene, and also the LacA gene. And you should notice that there is one more structure outside the Lac Operon, and this is actually a gene known as the regulatory gene, also known as the LacI gene. This gene codes for the repressor protein. And so if there is no lactose in the environment of the bacteria, there will be no need for the bacteria to produce the three enzymes just now. So in the absence of lactose, this will be happening in which the regulatory gene or the LacI gene is expressed, and this will produce the repressor protein. The active repressor will bind to the operator over here. As the repressor binds to the operator, the operon is switched off. Why? Because this prevents the RNA polymerase from binding to the promoter. As a result, there will be no transcription of the structural genes, meaning that LacZ gene, the LacY gene, and the LacA gene will not be transcribed. And so, there will be no translation and there will be no synthesis of the three enzymes. It is okay because there is no need for these three enzymes to be produced. Why? Because there is no lactose in the environment of the bacteria. But what happens if lactose is present? And so, when there is lactose, some of the lactose will be converted into allolactose, and this molecule can be considered as the inducer for the Lac Operon, in which the allolactose molecule will bind to the repressor protein, and this will change the shape of the repressor protein and causes the repressor to become inactive. Once the repressor is inactive, the repressor protein will not be able to bind to the operator over here, and we say that the operon is now switched on. As a result, the RNA polymerase will be able to bind to the promoter, and transcription of the three genes will occur, meaning that the LAC-Z gene, the LAC-Y gene, and also the LAC-A gene will be transcribed, and this will be producing one single mRNA strand, because the three genes are transcribed as one single unit. And later, the mRNA will be undergoing the process of translation, and as a result, the three enzymes will be synthesized. The beta-galactosidase, the permease, and the trans-acetylase will be produced, and this is important because the three enzymes are the important enzymes that will be involved in the metabolism of the lactose. And so that is how the Lac Operon can be involved in controlling the expression of the genes in the E. coli.